Hello, everyone. Welcome to our 13th webinar series. Today we have Femi Gabriel Oyeniyi, a senior lecturer from Ekiti State Polytechnic, Isan-Ekiti from Nigeria, and he will give us a presentation about nutrient composition of honey samples harvested from the same hive across different seasons, a case of Ekiti State Polytechnic. I hope that I've pronounced this correctly. So, dear Femi, the floor is yours.
All right, I want to start by appreciating everyone. First and foremost, the leadership of this cost action, the BeSafeBeeHoney. Thank you so much. I don't take it for granted, Dr. Andrea Freitas, Dr. Caroline Nabot, the manager whom I've been corresponding with over the last couple of weeks. I want to thank everyone present at this webinar. It's not lost on me that essentially the cost action is largely European in scope and in nature by virtue of the Horizon 2020 support. But the fact that I've been accepted to be a part of this, I don't take it lightly. And so I'm very grateful that I'm presenting these reports, which was born out of my experience when I helped to set up the apiary at my institution. And so without much ado, I'll go into the presentation right away. Just a minute while I share my screen, please.
Mm-hmm.
Please, can everyone see my screen?
Yes, we can see.
Oh, great. So the topic says nutrient composition of honey samples from the same hives. Pardon me to say, is not a single hive. There were, I will give the number of the hives that we used in the course of my presentation. So we did that across different seasons at the states, at the Polytechnic Teaching and Research Farm, specifically the Ekiti State Polytechnic. And this is important because the institution is relatively a young institution. It was just set up in 2020. That's just some 5 years ago. And so it was important that while we started, we were able to gather information, credible, evidence-based information, which I'm privileged to, some of which I'm privileged to share with you. So this is just the part of a work, a larger work that we did. And here is my presentation outline.
Sorry, Femi, can you just put, please, presentation mode on your presentation so it's a full screen?
Oh, great, great.
Yeah, that's it.
All right, is that okay?
Yeah, oh, no, now it's much better.
Oh, thank you so much. I'm grateful, NJ. All right, so we look into the background of this presentation, the materials and methods, and we talk about results and discussion, and as well as the conclusion and the recommendation. I forgot to add one more line, and that would be next steps, what we are looking at doing over time. As I mentioned, this was just a small part of a larger work that has been done since we set up that particular apiary some 3 years ago. So I begin with some background information generally. As all of us know, there are several things that influences the composition of honey at any point in time when honey is harvested. That has to do with the manner in which the honey was harvested, the seasonality, because this really talks about activities that happens in the hive itself. As we know, especially for hives that are reared in external environments, they are subject to weather and climatic vagaries. So season of the year, for example, during autumn, during summer, during winter, during spring, you have different activities of the bee itself or the bees themselves, and by implication, the honey that is produced. Also, one very important factor would be the sources of the flower, the kinds of flower, the species. So depending on the plant you're dealing with, maybe they are tree crops, maybe they are annual crops. Even with the annual crops, there are different kind of crops. We have legumes, we have grasses, we have all kind of forages. Okay, so all of those determine the composition of honey, which I believe we all know. It's not new to us. So I want to drive the point home to Nigeria. Nigeria, as we all know, is located in Africa, and Nigeria has got diverse agroecological conditions. So, for example, in some regions in Nigeria, we have a mountain vegetation. We also have forest vegetation, different kinds of forest. So we have the freshwater forest, we have the rainforest, we have the mangrove forest, we have the tropical rainforest, which is part of the zone in which I'm found, where this particular study was conducted. And we have the savanna and different kinds of savanna. There's a Guinea savanna, there's a Sudan savanna, and then the Sahel savanna. So for the Sudan and the derived and this Sahel savanna, they are largely semi-arid in nature. It's inching towards the northern part of Nigeria, which is getting close to the topmost part of the northernmost part of West Africa, and by implication is inching towards North Africa. And as you know, that is getting close to the Sahara Desert. And then we have the derived savanna. I'm saying all this to give the scope of what Nigeria looks like geographically and by means of vegetation. It's also important to state something that I think we all need to get accustomed to, or to know rather. Nigeria has the largest arable land in Africa, and this is significant. It's significant because, by reason of that fact, that country also has the largest population in the continent. I think the last I checked, even on a global scale, Nigeria is the sixth largest in terms of population, globally speaking. So it's quite a very large population. And as you can imagine, the demand for food will only rise over time. So a little bit of delving into how the apiary looks. So this plate that I'm showing here just gives a snapshot of harvesters on the way to the apiary unit. My apologies because the pictures didn't really come out clear, but you just see about some four four harvesters. They are on their way to the apiary in their bee suits. They also have their gloves well worn. Well, the face is not well captured, as you imagine. The person that captured it was also putting on the bee suit, so it wasn't very quite easy. And that's me actually. That's me in the second picture there. So we're on the way to the apiary to harvest honey, and this was done last year. So back to our presentation, I was talking about Nigeria. The significance of what I was talking about in terms of Nigerian vegetation is also appreciated better when we realize, and we know, I believe, that bees, honeybees specifically, because of course not all bees are able to produce honey. They are water bees, for example. But that's not the subject of our discussion this morning. I wanted to say that honeybees pollinate 75% of the world's flowering plants, and that's huge. That's significant, considering a nation like Nigeria that's got a huge population, and that population is projected to rise over the next couple of years, such that by the year 2050, Nigeria will be the third most populated country in the world, and a significant number of or portion of the food to feed that population will come from plants, as you can imagine. And as bees play a crucial role in the pollination of flowering plants, all of this is now brought into better perspective. But there was a report by the Federal Ministry of Agriculture and Rural Development some 5 years ago, some 8 years ago, sorry, that showed that the country has a potential of producing something close to 800,000 metric tons of honey annually. Though that has not yet materialized in terms of reality. So it's just the potential. As we speak, about a half of these, or even a little over a half, about 440,000 metric tons, are demanded outside the country, both within Africa and outside Africa. So Nigeria largely depends on imports. That's to say that honeybee plays a crucial role also for the Nigerian populace. And this is from a 2023 report just 2 years ago. As we know that Nigeria is a tropical country, the average temperature, even as we speak, this is the second season of the rainy season, or the second rainy season in the southern part of Nigeria. So there are two peak seasons of rainfall. The first starts from somewhere around April in southern Nigeria. It ends by, say, July. There's a little break in August, and then in September, it starts again. So that's the late raining season. And then it ends sometime in October or middle November. So as we speak, it's a rainy season in the southern part of Nigeria. But despite that, it's also hot all across the country. So it's it's a contrasting situation, so to speak. And this temperature is relatively so. This temperature, this condition, this tropical condition, holds for the greater part of the year, close to 9 to 10 months. I'm saying these to stress the point that at high temperatures, bees are generally less active, and that reduces honey supplies. But during low temperatures, very low temperatures actually, foraging also decreases because also during low temperatures, in temperate conditions, for example, it's winter, you see less plants that are flowering. And the activities in the hive are also relative, or sorry, the activities of bees in terms of foraging generally reduces. So there may be a need to supply the hives with food sources at this period. So it's important to then ensure that the hive is properly managed. And because we want to foster the welfare of bees, we're not just interested in having honeys. As they say, one should be interested both in the golden egg and the golden hen, the hen that lays the golden egg. And that's the case here when it comes to bees. We are not just interested in the honey itself, we're interested much more. And I believe this cost action has got several subgroups. There are groups that focus on the hive health and issues around monitoring toxicity in the environment, such that bees are not their health is not compromised. Okay, so a number of things such as wrong timing of harvest, over harvesting, even incorrect management of the hive may reduce honey quality, which is one of the focus of this presentation. So that then informed why this study was conducted. Among other things, we wanted to look at how specifically within the microclimate where the institution is located, if we could establish if there is a significant difference in the nutrient content of honey samples. So that was the first objective of the study. The second was wanted to ascertain if the period during which this study was conducted, this was last year, if it constitutes less stress. So I must say that the seasons were late dry season and then early raining season. So late dry season fell within March because the dry season usually starts from late November in southern Nigeria, either late November, depending on several variations, either late November or early December. And then it stops usually either late February or late March. So this study was particularly conducted in late March, and that was because we had obtained weather information from that locality to see when the late dry season ended before we then conducted this study. And then the early rainy season, it starts anywhere from April to May. So, but for this particular study, the early rainy season was during May. So the harvesting was actually done both in March and in May last year. It was conducted as I've mentioned earlier, at the apiary unit of the teaching and research farm at the Ekiti State Polytechnic. And the geographical coordinates are displayed on the screen. It's fairly high. As a matter of fact, the state where I am, where the study is based, is named after hills. It's surrounded by hills, actually. So the local name, Ekiti, actually means, in English, it means a city surrounded by hills. So it's about 544 meters above sea level, averagely. There are fairly larger points. I think the largest point is about 750 meters above sea level, but the average elevation is around 544, where this particular study was conducted. And so it's situated in the rainforest agroecological zone in southwestern Nigeria. And then honey was harvested from 4 out of the 6 top bar hives. We have top bar hives where what we used, what we still are using. So we have 10 out of those hives. Due to a number of issues, it was just 4 that we were able to harvest honey from on this particular occasion, both in March and in May, respectively. And we wanted to ensure that we were getting the honey from the same set of hives so as to be able to get the same or a similar result, right? That's why we ensured that they were the same. We also did harvesting usually in the morning between 8 to 10 a.m. Of course, we got to the apiary quite earlier, but we ensured that harvesting was done around this period. And then the product, the honey, of course, I mean, when I say the product, I'm referring to honey, the main product. Of course, there were other things that we got, but essentially honey, we tried to ensure they were placed in sealed buckets, very clean, hygienic. And what we did was a manual hand press at the Food Technology Laboratory of the Polytechnic. We then emptied, of course, we ensured that purification was done, labeling of bottles, new bottles, 33 cl bottles, centiliter bottles, that were previously unused. After we had completed the process of separation, allowing the honey to cool, to settle, we then put into these bottles and then sealed. 2 samples out of these during both seasons were taken to the laboratory, brought for proximity analysis and mineral content determination. We're interested in seeing what the content were. Though during the course of this study, we didn't look into details like to see if there were pollutants. To a great extent, we didn't look at that. Though we did check whether there was presence of lead, because we know that oftentimes lead is a huge contaminant in industrial areas. So we checked for the presence of lead, but for some other things like arsenic, for example, maybe contamination by some intense activities from the industries, we didn't check out for that, and it's something we intend to check as part of the next steps that I mentioned earlier. So these results that we obtained were then subjected to Tukey's test, since the results were obtained over 2 seasons. And in this particular slide, I'm keen on showing us a short footage of how the honey was harvested. Please pardon me because of the heavy size of the file. I tried to reduce it, and so it's not so clear. But what I'm doing is this slide I'm going to make available to Nemanja, and with my permission, it can be shared with the members of the group. So there is a link on YouTube where I posted the video where we can have a snapshot of how the harvesting was done. Okay, but I'll just try and show in the course of this little time without. So this is, or this was a caption, a footage of our visit to 2 of the hives. I mentioned that we harvested from 4 hives. So these were 2 hives located proximal to each other. So it could be captured in the video. But a clearer footage can be seen via the link, which I said after this meeting, we all can try to get access to. And so I move on to the results and the discussion from the study. We got a number of findings which we tried to pass together. We obtained weather reports as captured in table 1 and 2. Slide, and then the third table just compares the proximate composition as well as the mineral contents of both honey samples across the seasons. That's from March to May last year, respectively. So the first and the second table, I will just try and highlight the very salient points from there. For example, the average rainfall in the early raining season, that's in May, was 26.64 millimeters, and that was significantly higher than the one in the dry season, which was 1.5 millimeters. The wind direction also during the late dry season, that's March, was 205.77 degrees, and that was significantly higher or different from the former, which was 187.39 degrees. As for solar radiation, as we can imagine, during the late dry season, which is usually hotter, especially in the peak of the day, it was significantly higher than the early raining season. Also, table 3, and this is just a snapshot. I will show us the table shortly after this time. Table 3 also shows that the crude protein content of the honey sample harvested in the late dry season was significantly higher than the one in the early rainy season. It was 2.64% in the late dry season, and that was higher than the early rainy season, which was 0.69. So here's the table that I mentioned, but this is just for the late dry season. We see there the various parameters: rainfall, for example, relative humidity, wind speed, the wind direction, the solar radiation, and the relative pressure, respectively. So as we can see here, we have those same parameters for the early rainy season last year. As mentioned, you see the rainfall here is significantly higher than what you had in table 1. Also, you see the wind direction is significantly higher here. 205.77 is significantly higher than what you have during the early raining season. The relative pressure, there's no much significance difference. Okay, then we move on to table 3. I would just, as the last time, also try to pinpoint the very key and salient features of that particular table. So the crude protein, as I mentioned, was significantly higher, but that was not just what was higher. The potassium content of the honey harvested in late dry season was also higher. It was 101 milligram per kilogram during the late dry season, and that was higher than the one in the early rainy season, which was 19.35 milligram per kilogram. What this suggests to us is that there could be a higher proportion of nutrients. As we know, protein plays a very important role in wear and tear, in repair rather, of worn-out tissues in the body. As a matter of fact, about 50% of our body is made up of protein, aside water. 70% is made up of water, true, but for the hard matter, so to speak, is largely protein. Our fingers, for example, our hairs, and several other parts of our body, largely protein. So the macro molecules in our body are largely protein in nature. So what this means is that since we found potassium, for example, and we found protein and some other mineral elements in the honey sample that were harvested, for example, to be higher during the dry season, it suggests probably that there could be a correlation between a relatively low rainfall and the retaining of nutrients. And this is quite logical, as we imagine, probably due to the heavy content of water during rainfall that may have, to some extent, displaced what's it called, the viscosity. It may have reduced the viscosity of the honey, which accounts for probably why there was a higher content of protein during the dry season. That's what I just mentioned in the last slide. So the material was more nutrient dense in the dry season as compared to the raining season, which was also early. And so during May, we don't expect to experience a significant rainfall as against during other periods. For example, the peak of the rainfall is usually June and July, but then September, especially the last 3 weeks across the southern part of the country, in fact, maybe across the entire country in Nigeria, it's been heavy downfall, and this can be verified through the news. In fact, there's been cases of flash flooding across states, my state inclusive. Okay. So we then went on to interact with beekeepers. Ekiti is not known to have to be a high bee production state in Nigeria for a number of reasons, maybe. But when we interacted with beekeepers, we found that a swarming of bee colonies occurs between the months of March to June, with the latter being the peak period. So swarming is usually during June. And we also thought that this may also be responsible because during swarming, you also don't have so much of the honeybees around, and they separate from the colonies, okay? So maybe that may also have been responsible in part to what we had, since May is usually is closer to June than it is to March. This table gives the proximate composition and the mineral content of honey samples harvested across the different seasons. That's in March and in May, respectively. I'm sorry if it's not clear enough, but I'll just highlight a few things which I've done. The lead, I mentioned that we didn't really look for heavy metals and contaminants like arsenic, for example. But of course, we know lead is a heavy metal, so it was not detected as we see in the honey sample. And then as per the volume, we also found a significant well, maybe not so significant, but higher in the late dry season than during the early rainy season. I spotlighted others, for example, potassium, for example, protein. There are a few others that were slightly higher. For example, calcium. Calcium was also significantly higher, 222.65 milligrams per 100 gram during the late dry season compared to 98.75 during the early raining season. We also probably have, what again is that? Uh, Phosphorus. Phosphorus was 51.67 milligram per 100 grams during the late dry season, and then it was 22.25 during the early rainy season. And these are very important, what do we call it? Micronutrients that are needed. In fact, they are not micro, they are major they are micronutrients, but they are major micronutrients. You know, when you talk about micronutrients or minerals, they are macro minerals. I wanted to say they are macro minerals. Yes, they are macro minerals. So they are needed much more than micro, much more than trace minerals. Trace minerals, for example, molybdenum, manganese, magnesium, sorry, is a major mineral and is needed much more than manganese, for example. Phosphorus, sulfur are major micronutrients, and those ones are needed much more than micro. Iron is also another. So we could go on and on. And that just summarizes in a nutshell what we found out on the field, and what do those things that we found out, what do they mean? It then means that probably if the quality of honey and their products, propolis, bee bread, and others, may be maximized in this particular zone where the study was carried out, it may be advisable for farmers to schedule their harvest towards the late dry season. But we're speaking this not just for the local zone, maybe for the state. As I mentioned earlier, the state is Ili, but it's not even the state alone. So across the southwest, there is a trail of hills. So there are two major states that border these states. Osun State, for example, Ondo State, that hill or those hills also extends. The probable other exception to the hills would be Lagos, and Lagos is a coastal state. Okay, so even in Ogun State, which shares a large border, it shares probably the largest land border with Lagos in Nigeria. I want to believe most of us are familiar with Lagos. That used to be the former capital of Nigeria, and it's probably the most popular city people know in Nigeria, aside from Abuja, which is the capital city. So Lagos is part of the southwest. But Ekiti, where I am, is the farthest state from Lagos. It's about 800. It's about 300 kilometers away from Lagos. So that's about 7 hours of drive time, driving by road. By air, it's about a little over 1 hour, close to 2 hours by flight. Okay, I'm saying all these to mention that what we found here may be applicable. We are careful not to conclude, because there may be a need for validation for us to make an assertion and to make such a recommendation. But from what we found out, it appears that, and that's because this in itself was based on the study that we carried out last year. But don't forget that I said the APR itself started in 2022, 3 years ago. So we had started harvesting. The first year we harvested, we started June, the APR in June 2022, 3 years ago. And then the first harvest was in January 2023. So we harvested in January, we harvested in February, and we're harvesting. It then just occurred to me in 2024 that, wait a minute, why would we be harvesting something we just started that is new without getting data? Interestingly, I had just been accepted into the BeSafeBeeHoney group. It then occurred to me that I could go ahead to obtain data, and then at some point, which has now materialized, share those data with these distinguished scientists. And one of the things that I expect to gain is to look at points of, how will I call it now, collaboration. So things that we have not done so well, I want to believe, I mean, we've not had it so well. For example, there are a number of challenges which I didn't report in the course of doing this work and the course of running that hive. I also want to say that that's the first time in my life I was going to start an apiary. So this was a new area for me. And my background really is in animal welfare. So I was interested much more in the welfare of the bees than the products. Of course, the products were interesting, they are, and they still are interesting to me, but I'm much more interested in the welfare of the bees. So it was me just delving into insect welfare at this point in time. So as I mentioned, there's a need to validate the finding both within that particular area. So as I speak, data collection is also still ongoing. And when we are done with our findings, we will still publish the results at probably another opportunity. We also want to say that it may be good to then document what the harvest of honey in terms of the quality of the content looks like, and then other kinds of evaluation should be done. I believe that there are several other evaluations that this cost action has maybe perfected. So it will be glad for me to learn from other scientists how that can also be incorporated, and we could then have a kind of replication of methods and then see what the findings are. That really will be interesting. I know about the fact that there are some challenges also experienced in the Eurozone. So it will be more of a knowledge and experience sharing, which will better both continents. So better the Eurozone. It would also better the African Union at large. Once again, I want to thank this very eminent body of scientists for the opportunity to share a small part of my finding in the course of my work at Ekiti State Polytechnic. These are a few of the references that I cited, and I want to thank you for your very kind attention. I'll be glad to take questions as they come. Thank you, everyone.
Thank you, Femi, for this really awesome presentation. We have some questions. Let me just check. Hunter asks, was there any evidence that dry season honey had a lower water content that could affect the concentration of nutrients in the honey samples? Uh,
Oh, yes,
I think
thank
the
you.
speaker actually answered that in a later slide. I posted that before the relevant slide, I think. So I think you found there was a difference in moisture content, but not a huge one.
Thank you so much. Yes, yes. Thank you so much. So it was on the basis of the tables that, so we did a approximate composition, and I'm sorry, I didn't turn on my slide. My apologies. My apologies. I didn't turn on my slide. So when we did the proximate composition, and I can share the detailed report from the proximate composition if you don't mind. I can share that. The lab in which I did the proximate composition. So this can actually be verified. So it was on the basis of what we saw in the lab results that we reported. So thank you. I don't know if that answers the question. Otherwise, I'll be glad to share. My email is available, so I'll be glad to share the results just so you see. And thank you once again.
Okay, we have one more question from Gregory. What are the LODs for heavy metals?
Sorry, I didn't get that. What are the
LODs for heavy metals?
LODs? Could you explain what LODs means?
Gregory, could you explain also, please? I don't know what that acronym stands for.
Yes, sure. Hello, and thank you so much for this presentation. I would like to know what is the limit of detection for lead in particular, because you mentioned that there is no lead in your sample. So I would like to know what is your limit.
Oh, thank you so much, Greg. Greg, if you don't mind, I will be glad to be in touch with you after this presentation. I'm saying
sure.
that because, thank you so much. And I've made my email available already. So here's what I'm going to do. I'm going to link up with my my laboratory analyst, because I was not the one that ran the samples, and I'm going to request the procedure, and then I'm going to share with you so that this specific information, I'll be able to attend to it very much. But that information is not in my purview right now, but I would attend to it, and then I'll share that with you. If that's fine by you, I'll be glad to do it.
Okay, thank you.
Okay, we have one more question from Gordon. When in the year does the peak of the plant flowering season occur? at least of plants pollinated by honeybees in Nigeria?
This is a very interesting question. So, as you well know, that when you talk about plants and you talk about flowering plants, there are several flowering plants, right? Not all of them are annuals, not all of them are legumes. And then annuals, they even have their seasons. So, for example, I also want to use a very practical example, maize. Maize does about 3 months. In fact, less than 3 months. You have maize that grows within 75 days cycle right now. But look at cassava. Cassava is also an annual plant, though not a grass. It takes about 1 year, and it also has got flowers and several other crops. So they don't have the same cycle. So it will be difficult to pinpoint. But if I have specific examples of plants that you have in mind, I use maize because maize is a staple. Rice is also a staple, but oftentimes the kind of rice that we have is the swampy rice, and so you don't have them waiting much till the flowering period and what have you. But for the variety of flowering plants within where this particular study was done, maize is one of them, for example. Oil palm is one of them, for example. Banana, one of them, for example. And that would be during the raining season, usually when they flower. Okay, and so we have from June, or even earlier than June, you have May, for example, June sometimes, and then straight until September. But it's usually during the rainy season that you have this being done. Oh, thank you, Gregory. I just saw your email. I'll take note of that so that I'll be able to attend to the question that you said. Thank you. So back to the question that I was saying. So you usually have the flowering usually during that period that I mentioned. Thank you. I don't know if that question is answered, please.
Yeah.
Yeah, okay. I have one question, Femi, for you. How fast usually does honey in your country crystallizes? So I suppose, I mean, it depends on the flower, from which nectar is collected. But did you saw any, let's say, pattern of honey crystallization in your country? How usually does it fast becomes crystallized?
Thank you so much, NG. You asked me something I'm very interested in. I'm saying that because the honey that we produce, I also manage that. So we sell on behalf of my institution. And for several people that we've sold to, I've had at the last count about three, that reached out to me to mention what you just talked about. But what I asked each of them, and I requested for the sample of the honey. In fact, as I speak, I'm yet to get the result from the laboratory technologist, what those results are, because I was interested in finding what has happened. But I didn't um, tell them to check. I just said they should do a general content of what those samples were. I found out that the liquefied, or the, how will I say, the viscosity reduced significantly over time. What I then asked them is where they kept those bottles of honey, because what we packaged, the package that we used were plastic bottles in 33 cl containers, which were previously unused and were airtight and well sealed. And so, in almost all the cases, the three cases I mentioned, they put those things on the floor. So, I'm still trying to establish whether there's a correlation between bottles of honey, specifically the ones we use, being placed on the bare floor over a long time, or those that were placed on maybe tables or were placed in refrigerators, if there's a correlation between both. Because for other people, and we've sold, at the last count, we must have sold over 500 since we started. And I've not had this kind of reports apart from these three people, which I noted. So I can only speak about the ones I've experienced in person, which I'm still yet to get the reports from the laboratory technologists. But that's the much I can say about what I have personally done. Thank you so much.
Okay, thank you a lot, Femi, for answering that. Do we have any more questions? Uh, yeah, if we have none, I think that's it for today. Well, thank you again a lot, Femi, for taking your time out of your schedule for presenting us some of your work. And thank you everyone for joining us today. Hopefully, we will see each other next month. Thanks.
And NJ, just before you end,
Ah,
I'm trying to get Gregory's email. Could you help me send it to my inbox? I need to respond to his question.
okay,
Please help
don't
me.
worry. I'll
I'm
send
trying to
it
do
to
that.
you in the
Thank you.
email.
Great. I'm trying to copy it. It's difficult to do that. Thank you so much. I'm grateful.
Okay, don't worry, Femi. Thank you, everyone, and see you next month. Bye bye.
Bye bye.